Okay, these are the cell division and mitosis notes. You want to make sure you've gotten the note page from the chapter 11 drawer. If you haven't, make sure to go get it now. This is a little bit different than before. You want to make sure that you're listening because there are blanks on your notes that are also blanks in this PowerPoint that I will tell you. So you have to hear me say them, so you got to make sure you're listening. So we've talked a little bit about cell division already. Cell division is the process by which a cell makes two new cells. You can see it over here. We've got one cell making two, and then it makes two and it goes to four, and this is going to keep growing and growing. So when we divide, we take one and we literally divide it in half to make two. So why do cells need to do this? Again, we've hit on this too already. Cells need to grow. They need to grow us, really. Living things grow by making more cells. We just don't make bigger cells, we make more cells. We also need to repair our damaged tissue. When you get a cut, when you get hurt, we have to fix it. So our cells need to be repaired. And if our cells get too big, they're just not very good at getting the nutrients we need. They're not good at bringing things into the cells. They're not good at taking the waste out of the cells. When a cell is too big, it just doesn't work efficiently. So when our cells get too big, they need to divide so they can be smaller cells that work better. Now the original cell, the first cell, is called the parent cell. That's down here. You see that the first cell is the parent cell. Two new cells are called daughter cells. So this blank right here is just the number 2. Two new cells are called daughter cells. You can see right here, here are the daughter cells. We call them daughter cells, but that doesn't mean that they are female. Cells don't have a sex or a gender. So they're not literally females. We just call them daughter cells because these cells will become parent cells. And when we think about men and women, the only one of us who actually makes another organism are the women, right? They're the ones that carry a baby. So I guess the scientists decided to call these cells daughters because they become a parent who divides out. But they're not a female, they're just cells. Before cell division occurs, the cell is going to replicate, which just means copies, all of its DNA so that each daughter cell gets a complete set of genetic information. Look at our parent cell. We have one red and one blue chromosome. When we go to our daughter cells, they also have one red and one blue, but there's two of them. So when we went through DNA replication right here, we took one red chromosome and we made two. So remember, this is still one chromosome. Technically, it has two chromatids now. And the blue, we went from 1 to 2. And then when we moved, we split them. Now we have a red and a blue and a red and a blue, just like the original parent. Each daughter cell is supposed to be exactly like the parent, genetically identical. So they should have the same kind and the same number of chromosomes as the original cell did. So if you look at our parent cell, they had a red and a blue. The daughters, red and blue, red and blue. They are exactly the same. They're genetically identical. That's really good for things like our skin cells. They need our skin to be exactly the same. Now remember, our sex cells, our sperm and our egg that we do sexual reproduction with, those are not made this way. So our sex cells are not identical to us, which is why when parents have kids, the kids aren't identical to them. Our cells are different when there are sex cells, but the rest of our body should have identical cells. Many organisms, especially unicellular organisms, now think back to the word unicellular. Do you remember what that means? Look at that prefix. Uni means one, and then of course cell. So unicellular means one cell. So one-celled organisms reproduce by means of cell division. What type of reproduction is this? This is this blank right here. Do you remember? This is asexual reproduction. So asexual reproduction is when it's one organism splitting in half through mitosis to make two cells, which are now two organisms. If you're not sure how to spell asexual reproduction, look in your book or it's up on the standards on the board. Bacteria are one of our best examples of this. So bacteria, you only need one in order to reproduce. So this is how you get sick as easily as we can. If you just get one bacteria in your body and it lives long enough to go through asexual reproduction, you now have two bacteria in your body. And if those two reproduce, now you have 4 because all of them are reproducing on their own. That's how just one bacteria can get you sick, because they reproduce by themselves. Remember, humans don't do that. Humans are sexually reproducing. We need 2 organisms to reproduce. Let's talk a little bit about our DNA. So with our DNA, we know that DNA is located in the nucleus of our cells, and it controls all of our cells' activities, including cell division. But there's a couple different types of DNA here that I want to look at. So the first one is chromatin. Chromatin is the long thread-like DNA in a non-dividing cell. Look over here to this far left side. There's our chromatin. To me, it looks like spaghetti in a nice purple bowl, but it's really the nucleus with the DNA inside. So when the cell is in interphase, just kind of being a normal cell, it has chromatin DNA, meaning the DNA is just all loose and spread out. When we start to divide, we need to know where our DNA is. We need to organize it. So coiled short DNA is called a chromosome. Now this chromosome is actually the same as chromatin. Look at it. It's made of the exact same thing. It's just loose versus organized. And if you look at chromosome, is made of 2 parts, chromatids and the centromere. The centromere is in the middle here, and that is just where it's kind of held together. The centromere's job is to hold that chromosome together. The chromatids are each side of the chromosome. So if we're trying to explain differences here, chromatin is your loose DNA in a regular cell, not dividing. Chromosomes are condensed DNA, DNA that's been all put together and organized. And chromatids are each half of a chromosome. So right here's a little bit more explanation with chromatids. So chromatids are the pieces of chromosomes. You can have one, which is an unduplicated or a non-copied piece of DNA. And then when it gets copied, you just have 2 chromatids together. They're called sister chromatids because they're related. They're the same thing. If we just copy this guy, the other side is going to be the same as him. So we call them sisters because they are the same. And the middle is the centromere there, where they are being held together. One more time, because this gets really confusing. Chromatin is our loose DNA. When it copies, now we just have double loose DNA. But when it coils up, we make chromosomes. And why do our cells do this? Why do we split from chromatin to chromosomes? Well, it's just more efficient. Think about it. If our cells need to split our DNA perfectly in half, we don't want to lose any. When it's all loose like this, it's easy to get it jumbled up and confused and lost. If we don't want to lose it, we need it to be more organized, which is what it looks like in our chromosomes. So with chromosomes, every organism has its own specific number of chromosomes. So some of our examples is a human. A human has 46 chromosomes, or 23 pairs. Now pairs is a little bit new for us, but our chromosomes can come in pairs. What that means is that you got DNA, you got chromosomes from your mom and from your dad when you were created. Well, you actually got the same information twice. So our chromosomes are labeled by 23s. So your chromosome 8, whatever that is exactly the same as your other chromosome 8. If one says hair color, the other one says hair color. So while we have 46 chromosomes, we really only have 23 different types. We just have 2 of each. Dogs have 78 chromosomes. Goldfish have 94 chromosomes, and lettuce has 18 chromosomes. So I want you to look at these chromosome numbers here. With a human of 46, a dog of 78, goldfish of 94, and a lettuce of 18. What do you think chromosome number means? A lot of times we look at these numbers and we think that the more chromosomes you have, the smarter you are. But that is not actually the case. Look at it. Do you really think a goldfish is almost doubly as smart as you? They're not. Goldfish, just because they have 94 chromosomes, don't make them smarter than you. And lettuce, just because it has 18 chromosomes, doesn't automatically make it dumber than you. Is lettuce dumber than you? Probably. But not because of its chromosomes numbers, it's because it's lettuce. Our chromosome numbers don't really mean anything. It's kind of random. We have 46 chromosomes, but our chromosomes are packed with information, just full of it. Goldfish have 94, but theirs are just not as complex as ours. They're not as filled with information. The number itself doesn't mean anything to us. It's just literally how many chromosomes we have. Now, what is important on here, a little too far there, is when we talk about how our cells need to have the same kind and number of chromosomes. So here I'm talking about all somatic cells. Somatic is just a fancy word for body cells. So when I say the same number, I mean if a human has 46 chromosomes, which we do, then a skin cell of a human must have 46. A heart cell of a human must have 46. Muscle cells of a human must have 46. They all need to have 46 chromosomes if a human has 46 chromosomes. If you look over here, we have a picture of when all of our chromosomes are organized. So you can see there's 23 pairs, and there's 2 in each, which makes 46 total. This picture is called a karyotype, and we're going to talk about that in about 2 more chapters. Let's look at this fruit fly real quick. If a fruit fly has 8 chromosomes, then how many chromosomes will be in its skin cells, or its heart cells, or its muscle cells? You should fill those in. I'm hoping you knew to write 8 in each of these. If there's 8 chromosomes, then there's 8 in everybody. Let's go ahead and actually start talking about our cell cycle. This will go pretty quick. So cell cycle is a series of events, all the stages that a cell has to grow and divide. So a cell is going to grow, it's going to prepare for division, then it's going to divide to form 2 daughter cells. And this next part is why we call it a cycle to begin with. Think of the word cycle. Why am I calling this a cycle? Why am I drawing it as a circle? Well, because after we do all these things, we're going to begin the cycle again. So once it goes through the cell cycle, it starts again. Look right here. We start in interphase. We go around, get to mitosis, go to cytokinesis, and then bam, we're back in interphase, and the cells start growing again, and it all goes through the same process again. So your cells are always doing the cell cycle. That's why we call it a cycle. The first phase is interphase. Interphase is a period of cell growth and development. So it's kind of like normal time for the cells. They're just growing, they're hanging out, doing their normal cell activities most of the time. It's normal. So during interphase, we're going to grow and carry out our normal cell activities and replicate all of our organelles. But our cells are also going to do their DNA replication. They're going to copy their DNA. And the cell's going to spend most of its life in interphase. On average, and you might want to put this over here, 90% of a cell's time is in interphase, with only 10% saved for mitosis and cytokinesis. Now here at the bottom is a real picture of a cell in interphase. All you can really see is the cell membrane around the outside and this middle, which is the cell's nucleus. So the cell's nucleus and the cell's membrane. Now, real quick question, is this an animal or is this a plant cell? Well, it's an animal cell because it's a circle. Remember, our animal cells are circular usually, while our plant cells are always square or rectangular. We can only see the cell membrane and the nucleus. I don't see any chromosomes because there aren't any yet, not when a cell's being normal. It has chromatin. And I can't see any of the other organelles, but why? Well, it's really our fault. Our microscopes are just not good enough to see the other organelles. After interphase, we get into the specifics, really. So we have gap 1, synthesis, and gap 2. So gap 1, the cell's going to go through its normal functions. The cell's going to increase in size, and its organelle numbers increases. So it's being normal, it's growing, and I like to remember that right here. So when I think of gap 1, I match it with grow. So gap, grow. Synthesis, it says DNA is copied during this part. If you don't know already, let's learn real quick what the word synthesis actually means. Have you heard of the word synthesis before? Have we used it before? You might be thinking right now, well, it's part of photosynthesis, and you'd be right. We also might have seen it in our definition of ribosomes because they synthesize proteins. The word synthesis, and you may want to write this, synthesis means to make something. So when we synthesize, we're making something. So in the synthesis stage, what are we making? Well, we're making more DNA. The DNA gets copied, so we're making more. In photosynthesis, we made food from sunlight. The word synthesis is just to make something. Gap 2 comes after that, and the cell's going to continue to grow, and it does a final check before mitosis. So it makes sure that it's ready, that it did its DNA right, that it's grown big enough. It just makes sure everything's ready to go. So right here again, you want to say that the cell grows. So in both gaps, the cell grows. Now let's look at mitosis. Mitosis specifically is the division of the nucleus into 2 nuclei, each with the right number of chromosomes. So we're splitting one nucleus into 2. We're not actually splitting the cell in mitosis, just the nucleus and its DNA, its chromosomes. This is going to happen in all of our body cells. Remember, everybody but the egg and the sperm. So why does mitosis occur? Well, because we need DNA in both of our cells. Think about it. The DNA is the brain of the cell. It needs to be in both cells, and it needs to be there completely. If a cell doesn't have all its DNA, it's like it's missing its brain, and that would be really bad. So mitosis protects our cells to make sure it has a complete nucleus with all of its chromosomes. Here are the 4 phases of mitosis in order. Remember that we're supposed to think of PMAT. So here's prophase, metaphase, anaphase, and telophase. You'll be drawing each of these in just a minute, so you may want to pause the video and get some colors. You'd need 5 different colors. You don't have to color it, but I think it makes it a little bit easier to read, and these ones are colored, which makes it easier to know how to color it. Our first one is prophase. With prophase, the chromosomes coil up, meaning they actually are formed. They're not chromatin, they're chromosomes. The nuclear envelope disappears. That just means the outside of the nucleus here. So the nucleus itself is starting to disappear, and the spindle fibers form. These are new for us. This is a new organelle that we haven't talked about. The spindle fibers are literally just fiber. They're string almost, and their job will show up later, but they're to help the chromosomes move around. Chromosomes don't have energy. They can't move by themselves. So the spindle fibers help the chromosomes move. Now you should see on your notes that you're supposed to draw this picture, so you may want to pause now and draw it before you move on. Our next phase is metaphase. And with metaphase, I want you to think middle. So metaphase, middle. The chromosomes are going to line up in the middle of the cell. The spindle fibers are going to connect to the chromosomes here. So the spindle fibers help to move the chromosomes to the middle of the cell. You can see they've all lined up down the middle of the cell, and that makes them organized. It's all about organization here. We need to keep the chromosomes where the cell knows what to do with them and knows where they are so that we can split them properly. Again, make sure you're drawing. Our next one is anaphase. Anaphase, I think of apart or away. So anaphase, things move apart from each other. The chromosome copies divide. Remember, these were sister chromatids. The 2 blues were, the 2 greens were, they were sisters. They've been pulled apart, and the spindle fibers are pulling them to opposite sides of the cell. This is done because the cell's about to split right down the middle. We want the chromosomes to be as far away from that split as possible so we don't lose any chromosomes. So it's really important that they get pulled to opposite sides. The last one is telophase. With telophase, I think of 2 because we have 2 nuclei and we're about to have 2 cells. So the chromosomes uncoil. We are back with chromatin now. The nuclear envelope forms. It comes back, so we're getting a nucleus again. And the spindle fibers have disappeared. You can see this is not 2 complete cells yet because we haven't separated from each other, but we're really close. This separation happens in cytokinesis. Cytokinesis is the division of the rest of the cell, and that means cytoplasm and organelles. That's what goes here, cytoplasm and organelles. And that's done after the nucleus is divided. This looks different in animals and plants. And that's because the outside of animals and plants are different. Do you remember what organelle is on the outside of plant cells that are going to make it different than an animal? It's the cell wall. Remember, the cell wall was that hard structure on the outside of plant cells. So that's going to change how they divide. Animal cells get this little pinch. They pinch in and kind of pop off from each other. A plant cell can't do that. A plant cell just kind of grows a new wall down the middle. And they don't separate from each other because a plant kind of needs to all stick together while our cells move around our body sometimes. So an animal cell will pinch and separate. A plant cell will just form a new wall down the middle. And after mitosis and cytokinesis, the cell's going to go right back into interphase, and it's going to continue the cycle. I wanted you to see what real cells look like in cytokinesis, to really see this different. So the animal cell, it reminds me of like a half deflated balloon that's just kind of looking a little sad. It looks like a deflated balloon to me, where it's going to squish together, and it'll eventually separate. A plant cell's a lot more boring. It's just kind of building a wall right there. And once that wall's finished being built, it'll be 2 cells. So remember, in summary of the cell cycle, it's pretty simple. It's interphase, the 4 stages of mitosis, and then cytokinesis. And when it's done, it starts again. And remember, it only stops when cells die. So when cells become old or damaged, they're going to die, and they're replaced with the new cells. I wanted you to see what real cells look like going through all of the stages. So up here is interphase. You can see interphase as a drawing versus interphase in the real world. It really just looks like a circle with another circle. So it's the cell membrane with the nucleus here. Then here we have prophase. They're showing you at the beginning and when it's almost done. So you can see we went from chromatin to more like chromosomes. Here's metaphase, where it's lining up down the middle. You can see all the little pink spindle fibers coming at it. Here's anaphase, when it's just starting and when it's really going. Here's telophase. You can see it's almost 2 cells. And then here's cytokinesis, where it is 2. Real cells are not as pretty as our drawings, but you can see the point. You can see it lining up down the middle. You can see it pulling apart. So it matches, it's just not the same. Now, last thing I want you to do is I want you to look at this table. So this table kind of shows you everything about the phases, the important events, where the chromosomes are, what's happening. It kind of gives you the whole thing. This is really helpful. You don't have to write it down, but it might help you. You'll be doing something similar to this a little bit later in the unit when you do your lab. So you might want to remember this and come back to it when we get there. Your notes should be complete now, but you'll want to make sure that you complete the review page at the end of the notes. You'll turn that page in, so you'll pull it off your notes and turn that page in, and that'll be your grade for going through the notes. If you need a refresher on the notes, either read the ones you have, watch the video again, read the textbook, or ask me.